Hello everyone, I am Manimala Kadamanda and today I'll be presenting on dairy fermentation, production of yogurt. Yogurt is one of the most widely consumed fermented dairy products in the world. While many people know it as a healthy food, the science behind its production is a fascinating example of microbial biotechnology. In this presentation, we will explore how selected microorganisms convert milk into yogurt, the biochemical changes involved, the industrial products process, and the role of biotechnology in ensuring product quality and safety. Let's begin by understanding the basic principle behind yogurt fermentation. Principle of yogurt fermentation. Yogurt is produced through a process known as controlled lactic acid fermentation. Unlike natural fermentation, this process uses carefully selected starter cultures to achieve consistent quality and safety. The bacteria ferment lactose, which is the natural sugar present in milk, and convert it into lactic acid. As lactic acid accumulates, the pH of milk decreases from approximately 6.6 to 4.6. At this acidic pH, casein miscels become unstable and aggregate, forming a three-dimensional protein network that gives yogurt its characteristic thick texture. Apart from improving texture, fermentation also enhances digestibility, develops flavor, extends shelf life, and improves microbial safety. An interesting scientific fact is that casein reaches its isoelectric point at around pH 4.6, which is why coagulation occurs precisely at this stage. Starter cultures and their symbiosis. The production of yogurt mainly depends on two lactic acid bacteria. These bacteria exhibit a remarkable symbiotic relationship, meaning they help each other grow. Initially, Streptococcus thermophilus grows rapidly and begins producing lactic acid, along with compounds such as formate and carbon dioxide that stimulates the growth of lactobacillus. In return, Lactobacillus bulgaricus breaks down milk protein into peptides and amino acids, which provide nutrients that further support the growth of Streptococcus thermophilus. This mutual cooperation accelerates fermentation, improves flavor development, and results in high quality yogurts. Biochemical pathway of yogurt fermentation. Now let's look at the biochemical process involved. The lactose present in milk is first broken down into glucose and galactose. Glucose then enters the process of glycolysis, producing pyruvate. The enzyme lactate dehydrogenase converts pyruvate into lactic acid while simultaneously regenerating NAD+, allowing glycolysis to continue efficiently. As lactic acid accumulates, the pH decreases, causing casein proteins to aggregate and form a stable gel network. This biochemical transformation is responsible for the characteristic firmness and texture of yogurt. Industrial production process. Industrial yogurt production involves several carefully controlled steps. The process begins with standardization of milk, followed by homogenization, which prevents cream separation. The milk is then pasteurized at high temperatures, typically between 85 and 95 degrees Celsius. This destroys harmful microorganisms and denatures when proteins, improving the final texture. After cooling the milk to around 42 to 45 degrees Celsius, the starter cultures are added. The mixture is incubated for 4 to 8 hours, during which fermentation occurs. Finally, the yogurt is cooled below 5 degrees Celsius to stop bacterial activity before packaging and refrigerated storage. Each of these steps is essential for producing yogurt with consistent quality. Factors affecting yogurt fermentation. Several factors influence the efficiency of yogurt fermentation. Temperature is one of the most critical parameters because it directly affects bacterial growth and acid production. Incubation time determines the final acidity and texture, while limited oxygen conditions favor the growth of lactic acid bacteria. The composition of milk also plays an important role. Lactose serves as the substrate for fermentation, protein content influences gel strength, and fat contributes to creaminess and mouthfeel. Finally, the concentration and ratio of starter culture determines how efficiently fermentation proceeds. Maintaining these parameters within the optimal range ensures a consistent and high quality production. Texture and flavor development. The unique texture and flavor of yogurt are direct results of microbial activity. Certain lactic acid bacteria produce exopolysaccharides, commonly known as EPS, which increase viscosity and improve creaminess. Interaction between whey proteins and casein further strengthens the gel network, while reduced synthesis minimizes whey separation and improves product stability. Several compounds contribute to yogurt's characteristic flavor. Acetaldehyde provides the typical yogurt aroma. Diacetyl contributes buttery notes. Acetone adds a mild creamy flavor, and the lactic acid gives yogurt its refreshing sour taste. Together, these compounds create the sensory qualities that consumers associate with high quality yogurt. Quality control and safety. Quality control is essential throughout yogurt production. Physicochemical tests such as pH titrable acidity, viscosity, and syneresis help evaluate fermentation efficiency and product consistency. Microbiological analysis includes counting viable lactic acid bacteria, testing for yeast and mold, detecting coilform bacteria, and screening for pathogens to ensure product safety. Modern dairy industries also implement HACCP or hazard analysis and critical control points to monitor contamination risk throughout production. These quality assurance measures ensure that yogurt remains safe, nutritious, and consistent for consumers. Challenges in yogurt production. Although yogurt production is a well established process, it still presents several challenges. One major issue is bacteriophage contamination, where viruses infect starter cultures and delay fermentation. Another challenge is post acidification, in which bacteria continue producing acids during storage, affecting taste and texture. Syneresis or whey separation reduces consumer acceptance, while maintaining the viability of probiotic bacteria throughout storage remains difficult. In addition, strict hygiene practices are essential to prevent contamination by spoilaged microorganisms. Continuous research in biotechnology focuses on overcoming these challenges to improve both product quality and production efficiency. To conclude, yogurt production is an excellent example of how microorganisms can be used in biotechnology to transform a simple raw material into a nutritious and valuable food product. Through controlled fermentation, lactic acid bacteria convert lactose into lactic acid, result in protein coagulation, desirable texture, characteristic flavor, and improved preservation. The symbiotic interaction between starter culture together with optimized processing conditions and strict quality control ensures the production of safe, high quality yogurt. This demonstrates the significant role of microbial biotechnology in modern dairy industries and highlights the importance of fermentation in producing functional foods. Thank you so much for watching this presentation till the end. I hope you found it informative and gained a better understanding of the science behind yogurt fermentation. Thank you for your time and have a wonderful day.